Hello. In this video we'll be talking about um, insulin production. It's a technique of gene technology. Okay, so let's see what did the scientists do actually. So first we should know during the before 1980s. That's too large. Before 1980s, there was the insulin was collected from animals. Okay, so normally some people would not like the insulin from pigs or cows. They found it to be immoral or in such sense. Okay, and sometimes that insulin used to cause side effects, so it was even harmful sometimes. That was the reason that there was a great need to find a method to produce insulin by some other method. Okay, and this method was introduced in around 1970s and then developed completely in 1980s. So what did this method do? This method actually produced the exact kind of insulin as the one produced in the human cell, in the human body. The same insulin is produced. How is that done? We'll see in the following steps. Okay, so we'll have 4 steps. The first one is isolation of human gene, how to get the human gene, and then we'll have to prepare a vector, and then after preparing a vector we are forming a cloning, and we have to identify. Here identification is normally the use of markers. Okay, and formation of recombinant DNA. This is the we need a promoter for this one to occur, and then cloning, the reproduction, and finally we will come to the manufacture. So let's get into details. The first part is you take the beta cells from the human pancreas. So you know that these beta cells, of course, they will contain mRNA because there is constant production of proteins over here, right? When we studied in the regulation of insulin, we said that insulin, when there is less glucose, when there is more glucose, in beta cells in the pancreas are stimulated and they release insulin. That's what we said. So we are taking the same insulin. So obviously since they release insulin, which is a protein, they will have the mRNA, right? So we want this mRNA. Now what we do normally, what happens is from DNA, okay, we get mRNA, right? That's the normal process. mRNA. That's what happens normally, right? But now in this case, in the production, okay, maybe if we forgot, this process is called transcription, right? It's called the mRNA transcription in the protein synthesis. Now the reverse of this takes place in the production of human insulin. So this goes reverse. And how does it go reverse? There is this scientist got this virus which normally he does the opposite things. So they use this virus. Okay, it's called retrovirus. Okay, he has this reverse transcriptase. He has this enzyme. Okay, so this retrovirus have this enzyme reverse transcriptase. So this is used over here. So what do we get? We get a DNA from mRNA. Is that fine? Hopefully. Then after getting our DNA, So this one is called a complementary DNA, right? We can see here we took beta cells from the RNA from beta cells, some with the code for human insulin, right? mRNA with the code for human insulin identified and isolated. Okay, the one with the code for human insulin, as these days we can see the nucleotide sequence. We have the ability to see, so we identify the one with the code for human insulin and we isolate it, and then we sort of as we said, we add with the reverse transcriptase enzyme, okay, and we also add some free nucleotide, because once we get one strand of DNA, then it will pick the free nucleotides and it will form another strand. So from a single strand of RNA we get a single strand of complementary DNA, and then this complementary DNA undergoes replication, it replicates and forms two strands of DNA, of complementary DNA, okay? So this is the first thing. We are isolating human gene and we are getting our complementary DNA. Then this complementary DNA it does not end here. The process does not end here. We take this complementary DNA all the way to this side, right? So we took it here. So what do we have here? We have this complementary DNA. Now here it did not have sticky ends, you see? It did not have sticky ends, but we brought it here. So now in this case, in the second case here, this one have sticky ends. So we use restriction enzyme to break the points in the corners, so that we get sticky ends. Normally sticky ends means a region which can combine. Now we will need this for the next process, where we will be joining this DNA to a plasmid. So we will need a sticky end. And if you have the table of DNA showing the DNA, showing the RNAs, the triplet codes with the enzyme, you will see some stop codons and some start codons. Now those are the codons we want. We want to remove everything but those codons, so that we can eventually get it stuck in the plasmid in this over here. We will be able to stuck this over here, right? So we'll come to that later. First, let us finish this part of isolation of human gene. So mRNA is taken from beta cell and used to make DNA with the help of reverse transcriptase. It is then cut to leave sticky ends. Simple as that, okay? This is all the process of isolation of human gene. And the next process I'll be discussing in the next video. For the time being, hold on. Thank you for listening.